The illiterate of the 21st century will not be those who cannot read and write, but those who cannot learn, unlearn, and relearn. I believe you will be learned something about the cell cycle after watching this video. Hi everyone, this is John Vasanth James, Assistant Professor from the Department of Biotechnology, JJ College of Arts and Science, Pudukkottai. Humans follow a developmental pattern. They go through a series of developmental stage from the time of birth to the time they mature into an adult. And these transformations are mainly possible all thanks to the process of cell cycle and cell division. We all know that each multicellular organism begins life in the form of a single cell. So, how does a single cell get transformed into a large organism? To comprehend this, let's understand the concept of a cell cycle and cell division. In this video, we will be looking at the cell cycle. Let's begin with the definition. Cell cycle is a series of well-orchestrated steps including cell growth, duplication of genetic material, and cell division, usually resulting in two identical cells. In other words, cell passes through various steps in order to grow, copy its DNA, and finally split into either two or four new cells. The cell cycle was discovered by the Prevost and Dumas in 1824 while studying the cleavage of the zygote of the frog. In case of duration of cell cycle, it may varies from organism to organism and cell to cell. In case of human cells, it takes around 24 hours to complete one cycle of growth and cell division. Now let's look into the different phases of cell cycle. Cell cycle has two major phases, namely interphase and mitotic phase, that is M phase. Interphase lasts longer in duration compared to the M phase, and the major reason being that the interphase deals with the preparation for the actual division which occurs in the M phase. Interphase has three stages: G1 phase, that is gap 1 phase, S phase, and G2 phase. First, let us see G1 phase. G1 phase is the first phase occurring in the interphase, hence the name first gap phase. It corresponds to the interval between M phase of the previous cycle and the S phase of the current cycle. As the cell is metabolically active, it grows in size in the G1 phase, and all the cell organelles duplicate in this phase, and the building blocks which will occur in the later stages are made by the cell. Now let's move to the second stage of the interphase, which is the synthesis phase, abbreviated as S phase. As the name suggests, there will be synthesis of the most crucial part of each cell, that is the nucleus, along with the genetic material. It will get synthesized, so the S phase takes up the charge of the DNA replication. Now, along with nucleus, there is one more important organelle that gets duplicated in the S phase is centrosome. Centrosome contains smaller cell organelles called centrioles, which is usually placed at the right angle side. So, the S phase takes up the responsibility of duplicating the contents inside the nucleus and the centrosome present in the cytoplasm simultaneously. What next then? The next step in the process of interphase is the second gap phase, which is abbreviated as G2 phase. This phase allows the cell to grow more along with the enlargement in the cell size, and the cell also makes more protein and organelles. The most important function of this phase is that it prepares the cell for mitosis. This phase helps the cell to prepare for the actual cell division in the M phase. Let us have a look at the next important part of the cell cycle, that is the M phase. This M phase has four different phases, namely prophase, metaphase, anaphase, and telophase. Let us discuss all the four phases one by one in detailed manner. In prophase, the chromatin condenses to form the classical chromosome structure having two sister chromatids. Centrosomes move apart and begin releasing mitotic spindles. The nuclear membrane and the nucleolus also starts disappearing, which marks the breakdown of nucleus. After going through these changes, the cell enters the metaphase. The chromosome align at the equator, forming the unique metaphase plate. Along with this, the centrosome reach the opposite poles. Also, the mitotic spindle attaches to the kinetochore of the respective sister chromatids. All these changes prepare the cell to enter the next phase of the karyokinesis, that is the anaphase. The anaphase is the shortest and the most rapidly occurring phase in the complete karyokinesis. In this phase, the sister chromatids separate and begin their migration to the opposite poles of the cell. The cell then enters the last phase of the karyokinesis called telophase. This telophase carries out all the tasks reverse of what happened in the prophase. The chromosomes decondense to gain the relaxed string-like chromatin structure. The nuclear membrane and the nucleolus which disappear during the prophase now start reappearing. So now the cell is all set for the last phase of the mitosis, that is cytokinesis. The phase of the cytokinesis divides the cytoplasm of the cell. There is a formation of cleavage furrow in the cell membrane which extends up to the core, and this results in the formation of two daughter cells. These two daughter cells are genetically identical to each other. With this, we come to an end with the concept of cell cycle. Thank you so much for watching this video, and I hope this video is very useful to you. I conclude this video with my all-time favorite quote: the capacity to learn is a gift, the ability to learn is a skill, the willingness to learn is a chance. Thank you for your willingness to watch this video and learn some concepts from this. Thank you, friends.